is Bapak Ruby Setiawan, who will talk about metabarcoding analysis to assess the anthropogenic impacts on coastal marine microbial biodiversity.
Assalamualaikum warahmatullahi wabarakatuh. Good day, everyone. Thank you to the committee for organizing this seminar and the participants who attend this seminar. In this session, I would like to share our study on the marine microbiome entitled Anthropogenic Impact on the Diversity of Microorganisms Associated with Coastal Sediment in Tanimbar Island, Indonesia. Coastal areas have important ecological roles as habitats and are part of nutrient cycling for living things, especially humans. However, at the same time, they are vulnerable to the anthropogenic intervention and climate fluctuation. So they are often called the most dynamic ecosystem in the world. As the largest archipelagic country, Indonesia has been dealing with coastal water pollution. In addition, most cities in Indonesia are located in or near the coastal area, leading to increasing human activities, including agriculture, urban development, transportation, and etc. Those activities release pollutants into the coastal ecosystem and cause severe impact directly on the marine ecosystem and indirectly on human health. Microbes, which play a crucial role in nutrient cycling in the coastal ecosystem, are also vulnerable to the impact of water quality, altering their composition and functionality. Moreover, the pollutants are highly immobilized and accumulate in coastal sediment. This means microbes in coastal sediments might be a key factor in microbial response to the pollutant exposure over time duration. It is also worth to note that a change in microbial structure may affect the health of marine ecosystem. Hence, microbiome analysis combined with functional gene profiling can be used as biomonitoring and diagnosing tools for determining the ecosystem health from the microbiological perspective. Therefore, in this study, we investigate the difference in microbial composition and predict their functional profiles among sediment samples from coastal area in Tanimbar Island, Indonesia, using NGS based on 16S ribosomal RNA gene metabarcoding. The samples this study were taken between 17 to 18 April 2018 on Tanimbar Island, exactly in Saumlaki as high density population in HD and Lorurun as low density population or LD. The DNA extraction was carried out using Zymobiomics DNA mini prep kits. Then V3, V4 region of 16S RNA gene was amplified according to the protocol from Illumina. The DNA amplification was the PCR was carried out using MyTAC HS DNA polymerase, and the PCR product was sent to the sequencing company for indexing and sequencing. From a total of 10 samples, only 5 samples provide satisfactory sequence quality, 2 samples from HD and 3 samples from L. The obtained sequence were analyzed using QIIME2. The functional profile inference was performed using PICRAS2 with nitrogen metabolism selected from the KEGG orthologs. Ecological analysis and taxonomic plot were conducted in RStudio based on R version 4 using the vegan package. A total of 1,118 OTUs were observed from 5 samples and classified into 32 phyla, 71 classes, and 190 families. Proteobacteria and Firmicutes share a comparable number in HD samples. Meanwhile, Proteobacteria dominate the abundance of the LD samples. It's taken about more than 50% of the community, followed by Actinobacteria, Bacteroidota, and Planktomycetota. At the class level, gamma protobacteria was prevalent in HD and LD samples, accounting for about 22% and 50% respectively. A significant difference is observed at the family level, where Fibrionaceae predominantly in LD samples, and several families of Campylobacteria, including Sulfurimonadaceae and Sulfurophaceae, mainly observed in HD samples. The diversity index, Shannon index, and evenness index of the HD samples was higher than the LD samples. This means the microbial communities in HD samples were more diverse than in LD samples. Distinct separation was clearly observed in an MDS plot, where the LD samples made a separate group from HD. This might be happen because the microbial community structure was driven by soil organic matter concentration and composition. The gene related to assimilatory nitrate reduction and denitrification was completely found in both samples, where the assimilatory nitrate reduction is reduce the nitrate into ammonia, and denitrification reduce the nitrate into nitrogen. In the assimilatory nitrate reduction, gene near A, which convert nitrate into ammonia, was higher in LD than in higher in HD than in LD, and this contributed from Clostridiaceae and from Sulfurophaceae. Meanwhile, in the LD sample, it's evenly contributed from several families. Gene NASB, which convert nitrate into nitrate, was higher in HD than in LD. The Micromonosporaceae share the most NASB gene in HD sample, and also they are the only family that contribute to NASB gene in LD samples. In contrast, gene NAS-A and NAR-B, which convert nitrate into nitrate, was higher in LD and then in HD. The abundance of NAS-A genes was mainly correlated with the abundance of Vibrionaceae. And then the Archobacteriaceae was found to contribute to the NAR-B gene in HD. In contrast, Xenococcaceae gave a large proportion in LD after the chloroplast. And then gene related to denitrification was found higher in HD than in LD. The Bacilliaceae contributed to the gene near K both in HD and LD samples. The near S gene was dominantly shared from Sulfurophaceae in HD and the Marinobacteriaceae in HD samples. Furthermore, the Sulfurophaceae also contribute largely in the number of NORB and NOS-Z gene in HD sample, whereas Flavobacteriaceae enrich these two genes in LD samples. Composition of microbial communities shapes the ability of those microbes to utilize the available nutrition through certain metabolic pathway. And furthermore, the difference in microbial composition between urban and suburban coastal areas could be used as an indicator for coastal ecosystem management and pollutant severity levels. And to conclude this presentation, based on the predictive functional analysis, the perturbation in microbial composition occurs as a response to the nutrient availability in the ecosystem. Instead, the microbial change in the ecosystem can be used as an indicator of environmental health due to their ability to respond to pollutants. And thank you for your attention.
Thank you very much, Mr. Ruby, for very nice presentations regarding microbiomes in the coastal areas. We will move to the next speakers, which is Ibu Elizabeth Handini, will present about the effects of gamma radiations on orchids in Indonesia.
Good afternoon, ladies and gentlemen. Thank you for the time that has been given to me. I would like to introduce myself. My name is Elizabeth Handini. I am from Research Center for Plant Conservation, Botanic Garden, and Forestry. This time, I will present the result of our research entitled the effect of gamma irradiation on the growth of 3 Indonesian orchid species. For introduction, as we know, Bogor Botanical Garden is one of an ex situ conservation institution that has collected many orchid species, currently around 589 species that are included in the 106 genera located in the orchid house. Those species are potential to be developed as new varieties or hybrid of orchid for improving their characteristic as ornamental plants and for other purposes. The 3 species of orchid that we use for research include Coleogyna pandurata, known as black orchid, which shows by black lip of the flower, endemic to Borneo, and have a very short flower period, so it's typical for using breeding method. And second species is Payustan carpile, a terrestrial orchid that can live in both low and highland with a wide distribution and can be cultivated easily throughout the world. The uniqueness can bloom all year round and has a very striking color, potential as a medicine plant. But the flowers are always face down and self-pollinated. This behavior then can lead to the loss of seed. And the third species is Vanda trichala. This orchid species has potential as an ornamental plant with its fragrant flower, but the vegetative period is too long, so it takes time to bring out flower. Biotechnology through physical mutation using gamma ray irradiation has to be conducted to increase genetic variation. Therefore, this study aims to determine the proper doses that is able to bring out genetic diversity, which is expected to reduce weakness and increase existing strength with gamma ray irradiation treatment on the protocol. Methodology. This research was conducted in 2019 at the Bogor Botanical Garden Culture Laboratory, and irradiation process was carried out at Badan, which now become research center for radiation process technology. This study used material from the sport of orchid seed referred as protocom. The 1-month-old protocom was irradiated using gamma ray from cobalt 60 with dose 0 until 55 degree. Then transplanted to our subculture into specific media. For Coleogyna pandurata, we use facet and medium, Vanda tricolor we use chromor medium, and Payustan carpile we use Vibonec medium. The result of this subculture is called M1V1 generation. The life percent analysis was carried out by counting the life protocols after irradiation and then analyzed using curve expert 32 software to determine LD 20 and 50. Our results show that the protocol of Selugina Padrata and Tangarpile with those 30 and 50 gray could survive only for 12 months after irradiation. There's only one single protocol of Selugina Padrata which was irradiated with dose 15 gray that could grow. Meanwhile, all the irradiated protocols of Payustan Kerpile could not recover, and only the protocols that were not irradiated or controlled were survived. Of these three species, only panda tricolor can still survive, and analyzed using curve expert and found that the value of LD20 is at 32.35 gray and LD50 is 47.14 gray. Between these irradiation doses, there will be many potential mutants because the planet will have more diversity in those doses. Protocol irradiated with the dose 45 gray has shorter planet performance because of the shorter lived length, about 7 mm, that control planets about 20 mm. Also, the shoot at the dose 45 gray multiplied more than control, even though many plants no longer survive at this dose. A dose of irradiation 45 gray in panda tricolor give more shoot and shorter height of the plantlets. And the pictures are the result of gamma ray irradiation on protocol panda tricolor each 1 year and a half after treatment with 0 until 45 gray irradiation dose. And as a conclusion, mutation induction can be performed by using gamma ray irradiation. The doses of irradiation given for each species of plant which is capable of inducing mutation will vary depending on the radiation sensitivity of each species. Gamma ray irradiation dose above 15 gray are too high for Selugina Padrata and Payustan Kerpile. The lethal doses of panda tricolor were 32.35 for 34 gray on LD20 and 47.14 gray on LD50. Protocol of panda tricolor which were irradiated at dose 45 gray had shorter plantlet performance and multiplied more shoot than control plantlets. And the effect of gamma ray irradiation on the growth protocol will be specific for each species. The variation that will appear will increase the genetic diversity of the species, thereby increasing the biodiversity of the orchid. Thank you.
Okay, thank you very much, Ibu Elizabeth Handini, for presenting the effects of gamma radiation on orchids. We will move on our next presenter, which is Dr. Rafiq Sunaryanto. Dr. Rafiq will talk about the bioactivity of streptomyces against plant pathogens.
Good morning, ladies and gentlemen. I would like to present our research. The title is Streptomyces costa-ricanus RS25 with bioactivity against plant pathogenic. As we know that Indonesia is the largest palm oil producer in the world. Palm oil is one of commodity that is needed to boost the Indonesian economy. Palm oil contribute to the country largest foreign exchange in 2020 amount to 25.6 billion US dollar or around 358 trillion rupiah. Oil palm plantation and downstream industry open up many job opportunity with 3 million small and medium-sized enterprise, oil palm farmer, and involving around 16 million direct and indirect workers that can be absorbed. Ladies and gentlemen, behind the success of Indonesian palm oil industry, oil palm plantation are facing a decline in production due to attack by pathogenic fungi. One of the causes of the low production of oil palm is the attack of the basal stem root (BSR) brought on by Ganoderma brunense, and to that, almost 60% of oil palm plantation disease is caused by Ganoderma brunense. Cultural strategy as well as mechanical and also chemical management has been used to suppress BSR but have failed. Ladies and gentlemen, using resistant oil palm seed and also biological management method to combat this feature may be the most effective way to manage BSR. And also biological active compound or biocontrol agent are expected to be able to protect plant root of palm oil plant. The objective of research is to obtain terrestrial actinomycetes capable of producing an active substance that can suppress the growth of the oil palm plant pathogen Ganoderma brunensis. And next is material and method. Sample collection. Soil sample we took from Teluk Masjid, district of Sungai Apit, Siak Regency, Riau. 5 grams of each soil sample we take with sterile carbon tube at an average depth of 5 until 10 centimeters of ground level. And the next is isolation of terrestrial actinomycetes. Soil sample we was using sterilized water and then heat shock treatment and cereal dilution, and then we spread on humic vitamin agar and incubation at 30 degrees Celsius for 21 days, and then we will obtain the colony and we select the single colony. The single colony we preserve using cresol stock. The next is antagonistic activity assay. We use the dual culture assay, and the mycelial plaque of Ganoderma brunensis was placed on PDA petri dish, and then spora actinomycetes was treated in the same petri dish, Ganoderma brunensis face to face. The visualization is like this. This is the streak of spore of actinomycetes, and this one is mycelium of Ganoderma brunensis. And the next step is antifungal bioassay. Fermented extract is solved in methanol at 10,000 ppm. And at the same time, we prepare the Ganoderma on PDA. And the same time, also, we prepare paper dish were dripped with methanol solution of extract, and then we dry. And the last is placing over the paper dish on PDA agar surface media. And also the Next method is liquid culture for production of secondary metabolite. From glycerol stock, we inoculate the actinomycetes to YAME agar media, and then we inoculate again to the YAME broth, and then we inoculate it in fermentative culture. We use the minimal media. Fermentation was conducted during 7 days at 80 degrees with agitation 150 rpm. This one is extraction of active substance. We extract using ethyl acetate, and we remove the solvent using rotary evaporator. This one is a method for identification using molecular identification. The step is DNA isolation and amplification, and next is electrophoresis and DNA purification and DNA sequence. The data we put into the NCPH, and we have the data of species, and we try to make a phylogenetic tree. And the next is a result of our research. 60 isolate from 3 soil sample, we test to antagonistic activity again Ganoderma bonariensis, and this one is antagonistic activity isolate. This one is actinomycetes, and this one is Ganoderma. As we know, we can see that actinomycetes can inhibit Ganoderma bonariensis, and this one is antimicrobial activity. And this paper dish is containing extract of RS25, and we can see that the extract of actinomycetes can inhibit mycelium of Ganoderma bonariensis, and this one is methanol as a negative control, and also ethyl acetate as a negative control also. This one is a result of identification and visualization of morphology isolate RS25. The mycelium is like this. This is observation using microscope, and this one is a colony on agar medium in petri dish, and this one is observation morphology isolate RS25, size of medium, surface colony is rough, texture powder, egg of colony opaque, elevation is elevated, color spore is gray-white, mycelium substrate is yellow-white. And this one is the result of identification using genetic or molecular identification. Our isolate is similar with Streptomyces costa-ricanus strain K21C32. The homology is 100%. This one is phylogenetic tree. Our isolate is close related with Streptomyces costa-ricanus and also Streptomyces lanensis. The last is a conclusion. From this research, I conclude that one is 16 isolate of actinomycetes from 3 soil sample. One isolate is RS25, salt inhibitor reactivity on Ganoderma bonariensis. And the second, from the identification using genetic identification, our isolate is Actinobacteria. The genus is Streptomyces, and species is Streptomyces costa-ricanus strain K21C32, and homology was 100%. And the third is from phylogenetic tree, our isolate is close related with Streptomyces lanensis strain FORE-H3. Thank you very much for your attention.
Thank you very much, Dr. Rafiq, for the very insightful research on Streptomyces bioactivity. Our next speaker will be Dr. Andri Frediansyah, who will show us about the antioxidant properties and also antibacterial activity of Syzygium polycephalum. time is yours.
Hello everyone, good morning. My name is Andrew Ferdiansa from the Research Center for Food Technology and Processing, Badan Riset dan Inovasi Nasional in Yogyakarta. I'm going to present our research at the International Symposium of Innovative Bioproduction Indonesia on Biotechnology and Bioengineering with the title of Antioxidant Properties of Syzygium polycephalum leaf extract and its antibacterial activity against Serratia marcescens and Pulcholaria species. And the outline today has a 5-part. First, introduction. Second is the goals. The third is the workflow and method. And then the result and discussion. And the end is conclusion and outlook. Polycephalum belong to Myrtaceae family, and the tree can reach at height of 8 to 20 meter with stem diameter and crown diameter up to 50 centimeter. It is commonly found as a wild tree in secondary tropical forest at altitude ranging from 200 to 1,800 meter, but it also cultivated in residential garden as a fruit tree. This species is mainly found in Indonesia, especially in Java, Kalimantan, and Sulawesi, but it's also found in Malaysia and the Philippines. And there are many indigenous names for polycephalum, which is kowok, puruhi, kupa, domjong, gosem, and kaliasem. And the polycephalum ripe fruit is usually eaten raw as a rujak or as a candied. And young leafy shoot and sprout are eaten raw as a lalap, which is a raw vegetable salad with sambal terasi and eaten with rice. Polycephalum has a number of biological properties, including anti-cancer, antioxidant, and antibacterial activity against Staphylococcus aureus, according to the recent research. However, no study on polycephalum as an antibacterial agent against opportunistic pathogen have been conducted. Patients with severely weakened immune system may become infected with multiple opportunistic microorganisms at once. Pulcholaria species cause cystic fibrosis in human, and Serratia marcescens is a nosocomial bacterium that causes hospital-acquired bloodstream infection and pneumonia. Patients with weak hospital-acquired blood system infection and pneumonia can have benefit from the discovery of natural extract that capable combating both pathogen. And the extraction solvent is one of the most important factors in determining how efficiently bioactive molecule can be extracted from plant material. And previous research extracted bioactive molecule from polycephalum using various solvents including chloroform, dichloromethane, ethanol, and methanol. And therefore, no previous study have compared the effect of various common solvent on the phytochemical composition, antioxidant properties, and antibacterial activities of polycephalum. Thus, our goal is to determine the effect of two different solvents, which is methanol and 70% ethanol, on the extraction efficiency of bioactive molecule from the leaf of polycephalum, including total flavonoid, polyphenol compound content, and antioxidant capacity, as well as antibacterial activities against two opportunistic pathogens, in order to identify the best solvent for further extraction and research. Additionally, the second one is scanning electron microscope were used to visually demonstrate how the solvent used in the study could change the shape of the polycephalum leaf extract. And our research workflow, including extraction and evaporation, total polyphenol and total flavonoid determination, antioxidant determination using GPPH and ABTS, antibacterial activity, and scanning electron microscope, and statistical analysis. And the extraction began with mixing 30 grams with 240 milliliter solvent and measured twice at room temperature before being evaporated using rotary evaporator. The crude extract was then used for forest tests, including TPC using Prasetya et al. 2020, and also TFC using Triviono et al. 2022, and also DPPH using previous hour method. APTS also used Prasetya 2021, and antibacterial test using Chow et al. 2020. And finally, the same scanning electron microscope was carried out using Hitachi SU3500. And from the research, the results show that the total flavonoid content of polycephalum leaf extracted with methanol had a higher concentration than the extracted with 70% ethanol. However, the polyphenol content that was yielded by polycephalum that was extracted with methanol was much lower than that which was yielded by polycephalum that was extracted with 70% ethanol. And the second one is the antioxidant activities measured in DPPS and ABTS from polycephalum extracted with 70% ethanol were significantly higher than measured in leaf extracted with methanol. In earlier studies, the researcher found that the extraction solvent had substantially influenced on the antioxidant capabilities of variety of plant, which is from Allotman et al. 2009 and Michelis et al. 2012. And from this slide, the 70% ethanol extract was purple in color and resembled glass crystal, in contrast to methanol extract, which is only green in color and resemble granule. And it is possible that the potent antioxidant anthocyanin is responsible for giving its color similar to purple. Moreover, the granule-like appearance was noticed under the scanning electron microscope in the methanol extract, and the appearance of glass fragment was seen under 100-time magnification. And it is possible that the degree of antioxidant capacity in polycephalum leaf is contributed to the phenolic compound. This is since the ethanol group has large yield of phenol than flavonoid, and the anthocyanins are primary phenolic glycosides that are derived from the anthocyanidin. And distinct bioactive molecules secreted by different plant tissue each contribute in their own unique way to the overall antioxidant capacities. And the second experiment is the antibacterial activities against Serratia marcescens and Pulchoderia were significantly affected by extraction solvent. And according to the finding, polycephalum leaf methanolic extract had a lower MIC or strong antibacterial activity against Serratia marcescens and also Pulchoderia species in comparison to its ethanolic extract. And to the best of our knowledge, this is the first study to look into efficacy of polycephalum extract leaf against opportunistic pathogen, which is Serratia marcescens, as well as Pulchoderia species. And in the current study, various polycephalum leaf extract were tested for total flavonoid and phenolic content, antioxidant capacity, and also antibacterial potential. And the total flavonoid content of polycephalum extracted with methanol is greater than the polycephalum extracted with 70% methanol, 70% ethanol. However, the total polyphenol content is different, and the antioxidant capacity of polycephalum leaf extracted with 70% ethanol is outperformed that extracted with methanol. And moreover, polycephalum leaf extracted with methanol outperformed in Serratia and Pulchoderia antibacteria. And the result of the study showed that the different polycephalum leaf extract have distinct antioxidant, antibacterial, bioactive properties, and the further in vivo research into the antioxidant and antimicrobial activities of various polycephalum leaf extract for variety of useful application is required. And the plant extract studies could be used for food preservation in the future, as well as the nutraceutical, pharmaceutical, and natural therapeutic intervention for infectious disease treatment. I think that's all of my presentation, and thanks for your attention, and terima kasih. This is our group in PRTPP BRIN. Thank you.
Thank you very much, Dr. Andri, for your very interesting talk about Syzygium polycephalum. Okay, we'll move on to our last but not least speaker today, the sixth speaker of this parallel session, which is Ibu Mariana Susilowati. We're presenting about the morphological variation of clothes from various regions in Indonesia.
Thank you, moderator. Assalamualaikum warahmatullahi wabarakatuh. My name is Mariana Susilowati. I'm from the Center for Horticulture and Estate Growth. In this opportunity, I would like to present our research about fruit and seed morphology variation of clove from various regions in Indonesia. We know that clove is a Myrtaceae family and native crop from Moluccan Island, Indonesia. This crop has many important benefits, especially for cigarette, pharmaceutical, and pesticide industry. In addition, this crop can be used for COVID-19 therapy. Indonesia has clove production center in various regions, and the majority of farmers cultivate clove to meet their daily needs. So it is indicated that Indonesia had a great potential for clove development. But national clove production in Indonesia is still low. There is many factors affect this condition, one of which is the quality of clove seed. In clove, there is a high cross-pollination level, so that more genetically diverse of seed clove, even though it's originate from the same tree. Several attempts to increase national crop production. The first, extensification by increasing crop area. The second effort is using clove superior varieties. Indonesia has 5 clove superior varieties, and 1 clove superior variety was just released in 2022. The use of clove superior varieties must be supported by the good quality of clove seed. So it is necessary to observe fruit and seed of clove, because fruit and seed morphological variation can be effect on the growth and production. The objective of this research was characterize seed progenies derived from 83 selected clove high-yielding mother trees from 12 regions in Indonesia. This research used 83 humid clove derived from 12 regions in Indonesia as material genetic. This research was conducted at Greenhouse Indonesia Spice and Medicinal Crop Research Institute Bogor from August 2018 until April 2019. The observation divided into quantitative characters and qualitative character observation, and data analysis using STAR software. Data was analyzed using from descriptive statistics until correlation with tests. This is the result and discussion. In for quantitative character variable, we can see that the fruit length was relatively homogeneous with CV value of 2.45% until 12.27%. Based on CV value, Carlos Himid's fruit length was the most homogeneous. In the field, the carrot plantation is isolated from the others of population. So pollen contamination with other clove was very low. Humid from alors had the longest and widest fruit with CV value 6.29%. The fruit width was still relatively homogeneous with CV value less than 10% except Colaca's humids. The observation showed that the seed size was more varied than fruit size. Humid from Semarang and Inde had the long seed, and humid from Semarang and Alor had a broad seed and significantly different with humid from Trenggalek, Manggarai, and Colaca. The weight of seed and fruit flesh was main component determining fruit weight. Humid from Alor had the heaviest weight of fruit flesh, and included the heaviest seed weight group based on observation. This is the qualitative character variation of humid fruit and seed. The fruit shape divided into obovoid and oblong. Humid from Solo had different fruit shape, namely narrow, wide, oblong. The most of humids had fruit base shape was rotundate. Only humid from Cianjur, Manggarai, and Colaca had base shape was obtusus. All humids had fruit apex shape was rotundate with skin color grade purple group. The skin color of the majority of seed divided into 3 groups with varied in intensity of the red color from weak to strong. The shape fruit varied from short wide oblong to obovoid oblong. The skin color of fruit depends on maturity, so there is not much difference between clove from different regions. At the dissimilarity level of 17.4%, all humid divided into 3 groups. Group 1 consisted of humid from Cianjur, Alor, Inde, Semarang, Bogor, and Banggai Kepulauan. Group 2, only 1 humid, namely Colaca's humid. Group 3 consisted of humids from Trenggalek, Manggarai, Solo, and Karanganyar. This is the result of the correlation between fruit and seed morphology with plant growth seedling. The plant growth seedling parameter consisted of plant height, number of leaves, stem diameter, and number of branches on 8 months old after seedling. Based on person correlation test, no association between fruit and seed size with plant growth seedling parameter. This research concluded the quantitative character variation of clove fruit and seed from various regions in Indonesia range between 2.45 until 17.49%. The humid fruit and seed produced by Alor progeny were more significant than those produced by other humids. In general, the clove fruit was obovoid in shape and included bright purple group. At a dissimilarity threshold of 17.4%, the clustering analysis revealed that humid from various regions were separated into three primary groups. The last conclusion, according to the correlation analysis, there was no association between fruit and seed size with seedling growth. The result was funded by PTHM Sampurna Tbk in partnership with Indonesian Spice and Medicinal Crop Research Institute. We also thank to Mr. Turyatna, Mr. Rudiana Bakti, and Mr. Ramdan Arismaya for technical assistance at Greenhouse during this research. Thank you very much for your attention. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you very much for Ibu Mariana Suslowati for presenting her work on the cloves. So we have all of the 6 presenters presenting their results of their research, and we will move on to the question and answer sessions. So if you have any question to the presenters, please you may raise your hands or write any questions in the chat room. Is there anyone who would like to ask any questions or give comments to the speakers? No one? Okay, then I have several questions actually. So, okay, so we have here Dr. Tika, or she raised her hand. So yes, your time is yours.
Yeah, thank you, Pak Danang. So I have a question for Pak Ruby. I have several questions. So for this experiment, you previously mentioned about the significant equality about the sequence. So what do you mean by that, and how do you check the quality of the sequence? And then the second question is about the microbiome itself. You presented about the diversity of the microbes, etc. Do you also tested the significance in the microbiome of that what HDL and algae? I don't remember what is algae and HD actually. So do you also tested the significant in microbiome, like what the species, for example, or the genus? Because I did not see any genus mentioned in that figures. And then did you also test in the OTUs level? Because I saw only until the family, if I'm not mistaken. And also in the NMDS plot, you mentioned different cluster, and then you also mentioned about the soil organic matters. Did you also test the soil organic matters, like from the soil itself, or it's only based on the functional profiles that you check? So that's actually, I think that's all. Oh, also one more thing you mentioned about the chloroplast. I'm not sure if the chloroplast is microbes, because I think chloroplast is part of the microalgae part, and normally we remove that chloroplast from the analysis. Or you have any other reason why you put chloroplast in this analysis? Thank you. Thank you, Badan.
Thank you very much, Latika, for your questions. Is there anyone who would like to ask? If not, I'll let Mr. Ruby to answer the questions from Latika.
Okay, thank you, Latika. Sorry, but I can catch up with the first and second question.
Latika, could you please repeat the first and second?
But I will answer the third and fourth question first. First one, about the NMDS plot, there is a clear separation between two sites. We determine only based on the diversity of the microbes. We didn't conduct the soil chemical analysis because limited budget on that time. And then for the chloroplast, in the microbiome analysis, we remove the chloroplast, but in the functional analysis, because when we look at the functional analysis, it is a pathway, a complete pathway, which one contribute to that pathway. So the possibility of the microalgae contribute to the pathway is possible and also detected in the samples. Maybe you can repeat the first and second question.
Yeah. So for the first question is the satisfactory quality of the sequence, like how did you test that and how you remove, how's the filtering parameters, for example. Also with the second one is the significance in the microbiome, whether you test the significance which microbes in species level or in OTU level, not
Okay.
only in family level.
Yeah.
Yeah.
Okay. For satisfaction, the good sequence quality, we filter it based on the Q score. We use the Q score 19 as a threshold. If there is a sequence have a quality score below 19, it will be removed. And then in the taxonomy analysis, we only choose up to family level because the primer that we use, V3, V4 primer, is not a good primer to describe up to species level, because it's only a fragment about 300 base pair. It cannot uncover the species level. So several OTU are identified up to species level, but it's not many OTU. So we choose only in the up to family level, because the coverage of the those region, V3, V4 region, covering a good quality of distinction in family level. Thank you, Annie. Okay, thank you. I will give back to the moderator.
Okay, thank you, Pak Ruby.
Can I have one more question, Pak Danang?
Okay, sure.
Just one more,
Yeah.
about the primer that Ruby mentioned. So Ruby mentioned about the primer feature V4. Like, do you also have a suggestion, like which primer should we use to get more to species level, for example? If you have any idea. I mean, the V4 feature V4 is normally used for microbiome, right? So
several paper use V1, V2. It's have a depth taxonomy level up to species level, but the coverage is lower than V3, V4. My suggestion for the best analysis for microbiome is using full length 16S using long read sequencing. It will be better to conduct a study of microbiome, or maybe a shotgun sequencing.
okay, I think that's enough. Thank you, Pak Danang. Thank you, Pak Ruby.
Thank you very much, Ruby and Mbak Tika, for the nice discussion. I think we need to agree that studying microbiome with current technology, especially MySeq, for targeting very short sequences, are not very really suitable to detect until a species level. And I agree with Mr. Ruby, who suggested that we need a longer sequence technology to detect to the level of species. Thank you very much for the nice discussions. Okay, so we have another participant who would like to ask questions. Please. Can you unmute your mic? Okay.
Okay, Mr. Danang, thanks for the opportunity. My question is to Miss Elizabeth. In the conclusion, you state that the use of 45 gray of irradiation on the panda tree color, so the more should compare with the other plantlets. My question, is it no effect of irradiation by using this doses because you still obtain more shoots compared to the other treatment? Thank you.
Thank you. This is from Ibu Siti, right? Yeah.
Yes.
Okay. Okay. Thank you very much for your questions. Ibu Elizabeth, are you here?
Yes,
Okay.
I'm here. But sorry, I'm not catch up the question.
Ibu Siti, could you please?
Okay. In your conclusion, you say you state
Conclusion.
that the use of 45 gray of gamma irradiation on panda tree color so more suit compared to the control plantlets. My question, is it no effect of irradiation to this species? Because by using this treatment, you still obtain more shoots. Thank you.
More shit. Okay. I think we try to use the protocol to make what the radiation can make effect or to make the mutant. Yeah. We not use the plantlet in here. Sorry, is that
No.
answer your
I
question?
mean, in your conclusion, you state that by using the 45 gray irradiation,
Yes.
so more should compared to the control plantlet. You state in your conclusion. My question, is it no effect of irradiation? Because by using this doses, this plant is still produce more shoot.
More
This is the high doses 45.
We use until the 45 doses in here.
Yes.
And the effect of the performance of plantlet in that dose is make the multiplied shoot. Then the control is not like in that doses, not like 45 doses, I think. Or maybe it's
Okay, maybe you can answer in the next time.
okay. Thank you.
Thank you.
Sorry about my answer.
Thank you,
Okay, thank
Mr.
you,
Danang.
Ibu
Thank
Siti.
you.
Maybe you can. Okay. Yeah. You can talk with Ibu Elizabeth in a separate discussions to
Okay, thank you.
like the problem. Okay, thank you very much.
Okay.
Okay, do we have other questions? We have 7 minutes left. If you have any questions, please raise your hands. Otherwise, maybe I have several silly questions to ask, but hopefully interesting questions. Okay, if not, then I will ask to Ibu Ina. So you mentioned that there are several, Ibu Ina is here?
Yes.
Okay, so you mentioned that there are several tuber crops in the Moluccas. Have you tried all of them? How does it taste or that? Have you tried where the tuber crop plants? You mentioned that it's an alternative food source, right? Have you ever tried all of those tuber crops?
Sorry, my signal is not good, so it is. Yes, your.
Yeah, my question is. Okay, my question is: have you ever tasted the tuber crops that you presented before? I mean, in your food or something like that?
Oh yes. I also taste the tuber, and sometimes, like in a previous study in the Siawo and Sangye, we also make a food with people in there. So there is this species is what true that this is as an alternative food in there.
Okay, and the distributions, is it widely distributed in Indonesia, or is only distributed in the Moluccas, the tuber crop?
Some species it's widely distributed in other locations in Indonesia, and some species also restricted in that location.
Okay, thank you very much for your answers.
Thank you, Mr. Tenang.
You're welcome. Is there anyone who would like to ask questions to the speakers or discuss something about the speakers' themes? Now we have 4 minutes left. Maybe last questions probably to Ibu Mariana. I have small questions. So you mentioned about morphological variations of the clothes, the length and the width and so on and so on. Does it have anything to do with the quality of the clothes that it produces? For example, longer seeds produces better clothes, or shorter seeds are producing better clothes, or something like that? Does it have any correlation between the morphological variations of the clothes with the quality that it produces?
Thank you, Mr. Danang, for the question. I will try the answer. In this research, we observe quantitative and qualitative characters. Quantitative, fruit and seed, length and width, and weight. And qualitative character, we observe the colors and the shape. From the result of this research, there is no association between quantitative character of fruit and seed with seedling growth on 8 months after seedling, but we trust that fruit and seed morphological variation has associated with cloth production. In this research, image from alars has fruit with long and brought fruit and seed, and humans from others had a good performance of seedling. Thank you.
Okay, thank you. Thank you very much for your answer. Okay, so we will conclude the sessions, and as we have only 1 minute left for the sessions, I think we can move on to the main rooms. I would like to thanks to the presenters and also to the participants and also those that are asking the questions. So you may leave these breakout rooms and return to the main rooms, as we will have 2 interesting plenary sessions, which will be delivered by Professor Ivo Nemas from Germany and also Professor Claudio Tiribelli from Italy. So I think that's all to conclude these sessions. I think thank you very much again for everyone involved here. So, see you in the plenary sessions. Thank you very much. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Recording in progress.
Welcome back to the main room, ladies and gentlemen. The next agenda is plenary sessions for this afternoon. Therefore, I may invite Dr. Ira Handayani as the moderator of these sessions. But before that, allow me to read short profile of Dr. Ira. Dr. Ira Handayani is currently a researcher in Research Center for Applied Microbiology, National Research and Innovation Agency, the Republic of Indonesia, who are interested in molecular genetics, microbiology, and bioinformatics. She obtained her bachelor degree in chemistry at University of 11 Maret, and she continued her master degree in biochemistry at Bandung Institute of Technology, or ITB, and also her PhD in chemical engineering at Tübingen University, Germany. Dr. Ira, time is yours to lead the sessions.
Okay, thank you, Ms. Aulia. Are you hear my voice? Okay. Hello, are you hear my voice?
Yes, we can hear you
Okay.
clearly.
Okay.
Dr. Ira.
I don't know whether Yvonne already in here or not.
Yes, good afternoon, Ian.
Oh,
I hear you and I'm already here.
Okay, how are you?
Thanks, fine. Hope you too.
Okay. Okay, thank you. Okay, I will start the presentation. The plenary session for this seminar. First of all, I'm very happy to see you here and welcome all of you to this plenary session. Before the presentation starts, let me tell you how the presentation will go. First, our first speaker, Professor Ivo Nemas, will be invited to present the material, and then our second speaker, Professor Claudio Tiribelli, will also be asked to present his material. After both speakers present their material, there will be a question and answer session followed by a conclusion. Now let's start with our first speaker, Professor Dr. Yvonne Mas. Okay. This is the CV of Professor Dr. Mas. Professor Dr. Mas is a professor of applied microbiology at the Technical University of Brunswick and head of the Department of Bioresources for Bioeconomy and Health Research at the Leibniz Institute, the SMZ, a German collection of microorganisms and cell culture. She has experience as an editor in the journal Molecule and the journal Printer in Microbiology, and experiment as a member of the topical advisory panel in the Journal of Antibiotics. She is interested in research in the field of microbiology, biotechnology, actinomycetes, antibiotic, and regulation. Now allow me to welcome Professor Dr. Yvonne Mas to make her presentation. Professor Mas, please explain your material. And time is your Yvonne.
So thanks, Ira, for this nice introduction, and good afternoon to everybody. Okay, now I see my slides. Okay. Okay. So again, good afternoon to everybody. Yeah, thanks for the nice introduction. Actually, I'm here at the Leibniz Institute DSMC, so the German collection of microorganisms and cell cultures in Braunschweig, Germany. And it's a great honor to give a talk during your session at the International Symposium of Innovative Bioproduction in Indonesia. And today I would like to introduce you into the topic of how we can exploit the biodiversity for novel natural compound production by using regulatory switches. So I should, now okay. So before I go into that more in detail, I first want to mention why this kind of research is relevant at all. And I think all of you know that antibiotic research indeed is relevant because we observe growing crisis from antibiotic-resistant bacteria. So the World Health Organization regards the antibiotic resistances as one of the 3 biggest threats to global health. So of course, it's urgently needed to find novel antibiotics to combat these drug resistances. But the question is where to find them. I'm sorry, the presentation is a bit slow, but I hope we can do that.
I think it's okay.
Okay. So we can, of course, find antibiotics a lot in actinomycetes. So actinomycetes are known to produce 2/3 of all antibiotic in clinical use today. And this antibiotic production potential is known for quite a long time. However, what is not known for such a long time is that the genetic potential for the production of antibiotics is even larger in these organisms. And this appeared when the first genome sequence of such an antibiotic producer has been sequenced. So this was the genome sequence of the model organism, Streptomyces silicolor. And that silicolor strain is known to produce up to 2 to 4 secondary metabolites under standard lab conditions. However, the genome sequence analysis revealed that the genome harbors up to 20 different biosynthetic gene clusters. However, not all of those gene clusters are expressed, meaning not all of these substances are really produced, but a lot of them occur as so-called silent antibiotic gene clusters. And this is not only true for that organism, but nowadays, since we have a lot of, more than hundreds of genome sequences available from different actinomycetes, it turned out that this is true for a lot of these producing organisms. So obviously, actinomycetes still hold a huge potential for the production of novel secondary metabolites. And of course, the current trend is to activate these kind of silent gene clusters. And now I would like to introduce you into the topic, how we can activate those kind of silent gene clusters in actinomycetes. Okay, and we actually can do that by using regulators. And regulators indeed are the set screw to unlock the biosynthetic potential of these natural compound producers. However, the problem is just that there are so many different transcriptional regulators abundant in actinomycetes, especially in streptomycetes, and these can be of different classes, and they can interact in quite complex cascades with each other. And depending on genome sizes, those can actually be more than thousands of different transcription regulators. So the question is, which is the most suitable and most potent type of regulator that is suitable to be used as a general activator of antibiotic biosynthesis? And today I will focus on these subtype regulators, since we think that these are good candidates to be used as general activators of antibiotic biosynthesis. Okay, presentation is still slow, but okay. So first of all, I will introduce you where we found this type of regulators. And we indeed identified them in the antibiotic producer Streptomyces pristinus pluralis, which is kind of our working horse. So this strain produces the pristinamycin substance, the streptogramin antibiotic pristinamycin. And pristinamycin is actually two different types of antibiotics, the peptide antibiotic pristinamycin 1 and the polypeptide antibiotic pristinamycin 2. And the Streptomyces pristinus pluralis strain produces those two substances in a specific ratio. They interact synergistically with each other, and a semi-synthetic derivative of that substance is on the market. It's called Synergid, and it's used as an antibiotic of last resort against highly resistant pathogens. And what we did back then was that we characterized the antibiotic biosynthetic gene cluster. And back then, this was the largest antibiotic gene cluster that has been described so far. And the genes that you can see here in red, they are needed to, they encode for the peptide antibiotic pristinamycin 1, and the blue ones encode for the polypeptide antibiotic pristinamycin 2. So when we had a closer look into this antibiotic gene cluster, then we identified a set of 7 different transcriptional regulatory genes, and they belong to different types of regulatory families. And actually, this is quite a large number, because usually these kind of antibiotic gene clusters have 1 or 2 such kind of regulatory genes. And we studied the function of all of those regulators in detail. However, today I will only focus on this red-labeled PAPA2 regulator here, which belongs to the subtype family of regulators. So when we studied the function of this regulator more in detail, and there I need the next slide. Okay. First of all, we inactivated the gene and studied the pristinamycin production, and there we observed that the inactivation of the PAPA2 gene leads to complete loss of pristinamycin production in comparison to the wild type, which still nicely produced pristinamycin 1 and pristinamycin 2. So therefore, we knew that PAPA2 is an essential activator of pristinamycin biosynthesis. Ira, can I ask, are you the one who is moving the slides forward, or can I do that?
No, I don't know.
Okay,
no,
because it's.
I think it's a bit left in there.
Yeah, it's really several.
I will ask Sandy.
But I will. It's okay. I will just move on. So PAPA2 is an essential activator of pristinamycin biosynthesis. This was what I wanted to say. And we studied the function of that regulator also more in detail, and figured out where the regulator really binds within the antibiotic gene cluster. So therefore, we purified the PAPA2 regulator and did electrophoretic mobility shift assays, and thereby we could show that PAPA2 really binds to the promoter regions in front of the pristinamycin 1 and pristinamycin 2 structural genes. And we were also able to define a specific consensus sequence for such a binding. And by RT-PCR experiments, which are shown here at the right-hand side, we also could show that there's no transcription of the pristinamycin 1 and pristinamycin 2 structural genes in the PAPA2 mutant in comparison to the wild-type organism. So therefore, we knew that PAPA2 is a direct activator and an essential activator of pristinamycin biosynthesis in Streptomyces pristinus pluralis. And the next slide, which I hope comes in a few seconds, shows you where the regulator is situated within the regulatory signaling cascade. Because it's here located at the very bottom, it directly activates the pristinamycin biosynthesis, and itself it is regulated by a set of other transcriptional regulators where I will not go in detail. However, it's quite complex signaling cascade governed by microbial hormones called GABA-butyronolactones. And what I wanted to mention here is that understanding these kind of regulatory signaling cascades and the regulation mechanisms of antibiotic biosynthesis in general can also help to activate silent gene clusters. And this we observed when we overexpressed the PAPA2 regulator in a foreign strain. And back then, this was Streptomyces lividans. And this lividans strain should come also in a second. This lividans strain usually does not produce any secondary metabolite under standard lab conditions. So this you can see here on the left-hand side, the lividans strain with the anti-vector control. However, when we overexpress the PAPA2 regulator, then the lividans strain produces an intensively red pigmented substance. And this red pigment indeed is andecyl protegiasin. And andecyl protegiasin is a known product from Streptomyces lividans. However, it is usually not produced in the lab, so it's encoded by a kind of a silent gene cluster. And this andecyl protegiasin biosynthesis is known to be under control of a subtype regulator called RED-D. And this RED-D shows quite high sequence homology to our PAPA2 regulator, so up to 55% sequence homology. And therefore, we speculated that PAPA2 takes over the function of RED-D and leads to the activation of this silent andecyl protegiasin biosynthesis in Streptomyces lividans. And we also later on could show that on the molecular level. So I'm really sorry that it's hanging, but yeah, this is what is shown in here. So having that in mind, we had a closer look into this kind of subtype regulator families in general. So in literature, they are described as Streptomyces antibiotic regulatory proteins. They are known to act as pathway-specific activators of antibiotic biosynthesis. And all of them contain a conserved N-terminal DNA binding domain. And with this, they bind to conserved consensus sequences at the promoter DNAs of the promoter regions in front of the target DNAs. However, so far, the regulatory mechanisms, how those regulators really work, is totally unclear. So this is something what we are going to study in the near future. So we will do crystallizations of the regulator and try to figure out how this activation mechanism really works out in the antibiotic producers. So having that in mind, we had a closer look where those subtype regulators occur, and we found that the genes are very abundant in many different actinomycetes. So you can find them in nearly all Streptomycetes, but also in other important antibiotic producers belonging to the actinobacteria family. And when you have a closer look where they occur within the genome, then you can find them as part of biosynthetic gene clusters. And actually, as part of gene clusters that encode for different types of secondary metabolites, like beta-lactam antibiotics, phosphonate antibiotics, polyketides, and so on. So therefore, we knew, okay, the subgenes are abundant in very different actinomycetes. They are part of gene clusters encoding for different types of secondary metabolites, and therefore we said that those ones are good candidates to be used as general activators of antibiotic biosynthesis. And then we tested this out in a random strain in just a running project of ours. And this also will be shown, I hope, in a few seconds. Because back then, this was a project which we performed with some Indonesian partners. So actually several ones that are sitting in the audience today. So with Puspita Listianti, Wien Kusaryoto, Iran Dayan, Shantaratna Komala, and so on. So back then we aimed to isolate new antibiotics from Indonesian actinomycetes. So there we are. Back then, the Indonesian colleagues isolated up to 422 actinomycetes from Indonesian habitats. A subset of strains was handed over to our labs back then at the University of Tübingen, and we prioritized the strains for the most phylogenetically unique ones. We grew them in several different cultivation media and covered all of that to bioactivity tests to prioritize the one with the best bioactivity. And back then, 9 strains were of most interest, and from these ones we isolated the genomic DNA of, did biosequencing, and then performed a combined genome mining molecular networking approach. And I think this was what Ira mentioned some hours ago in her talk. So this we have done for dereplication purposes and focus on the most unique gene clusters that encode for the potential unique substances. And back then, one of the interesting strains that turned out from this analysis was SHP-227, which showed quite some good activity. And we just randomly used that strain and performed our self-guided activation strategy with that. So we cloned in our PAPR2 overexpression strain to SHP-227 and got extracts of the cultivation of the strains and tested it against Bacillus subtilis as a test organism, and as you can see in here, the SHP-227 PAPR2 overexpression strain showed significantly improved bioactivity against Bacillus subtilis in comparison to the control strains with the anti-vector or the wild-type samples, which means that PAPR2 overexpression led to significantly improved bioactivity of that SHP-227 strain. Now, what you would do in the next step is that you go into the lab and do elaborative isolation of that compound and check what the substance really is. But we didn't do that. In the first place, we started a genome mining approach of that strain in order to find out what might be the substance that encodes for this, or what might be the cluster that encodes for the substance with this improved bioactivity. And as I said, we had the genome sequence in hand. We performed an anti-smash analysis for the prediction of potential antibiotic gene clusters, and thereby we identified 25 potential secondary metabolite gene clusters. And then the question was, which is the one that encodes for this bioactive substance? And then we ran an additional bioinformatic analysis by applying the program PET scan and searched for our PAPR2 consensus sequence. And thereby we found this subtype sequence in front of gene cluster 9, and gene cluster 9 encodes for potential nucleoside antibiotic, which has similarity to a gene cluster from a Streptomyces lineaceus therapus strain. And that one is known to produce the substances amicetin and plicacetin, which are known compounds. They act as protein synthesis inhibitors with quite good bioactivities. And only when we knew that, then we went into the lab and checked by HPLC analysis what the product of this strain really is. This should be shown in the next slide, hopefully in a few seconds.
Sorry, Yvonne, I'm interrupted. Your time is 3 minutes again.
Okay, I will hurry up.
Sorry.
And I will go through the slides. Okay. So back then, we indeed could show that it's amicetin plicacetin that is produced by the PAPR2 overexpression strain and in significantly higher amounts. In the next slide, it is shown that this also helped for the isolation of for novel pyrimidine nucleoside antibiotics, which was a collaborative work with the group of Harald Groß at the Pharmaceutical Institute of the University of Tübingen. I'm really sorry, but the slides are just too slow. So this was just published in the beginning of this year. And now we know that this sub-activation strategy really works out. We are now having a set of different overexpression plasmids. I will skip that, but I hope, yeah, I hope that will work. So what we are just doing at the moment is that we apply this to the genome sequenced strains of the DSMC. So we scale that activation strategy up, and we already screened hundreds of genome sequenced DSMC strains for the abundance of sub-genes and sub-consensus sequences. And these are the ones that we are currently using for performing our sub-guided activation strategy. And in one of the next slides, hopefully, I still have the time that also, I will skip that one, that also in our selected DSMC strains, where we clone in our PAPA2 regulator, we observe significantly increased bioactivities in comparison to the control strains. So when we test different test strains as for the bioactivity. And for some candidate strains, we already have some potential substances that are produced. For several strains, you can already see by the eye that something is going on, that when you clone in the PAPA2 construct, that there's significantly production, or that there's production of a pigment in comparison to the wild type strain. And we are currently isolating these substances and trying to find out if there's a novel antibiotic that is produced by those strains. And with this, I'm at the end. I'm really sorry that the slides were kind of delayed, but I would like to thank all the people that have been involved in this project, mainly Ira Handayan, who worked a lot with these Indonesian strains, especially with the SHP27 strain. Also, Janina Krause performed a lot of these sub-overexpression analysis, and currently the right-hand person are involved in sub-guided activation strategy of the DSMC strains. And with that, I would like to thank for your attention. I hope we can move on with joint collaborations, because I really enjoyed that interaction with you. Okay, so thanks, and I would be happy to take questions later on.
Okay. Thank you, Yvonne. Thank you very much for giving such an informative and exciting presentation. Now, let me invite our second speaker, Professor Claudio Tiribili, MD, PhD.
Here I am.
Okay. Professor Tiribili. Okay. Okay. Wait a minute. Okay. Okay. Okay, thank you. Professor Tiribili is the scientific director at the Fondazione Italiano Fegato. He has
Italian Foundation. That's easier.
Oh, okay. Sorry. He has experience as an editor in the Journal of Hepatology and the Annals of Hepatology Journal, and experience as an associate editor in the BBC Research Note Journal. He is interested in research in bilirubin, neonatal jaundice, metabolic liver diseases, hepatocellular carcinoma, stem cell in hepatocellular carcinoma, Klicker nasal syndrome, and translation medicine. Now allow me to welcome our second speaker, Professor Claudia Tiribili, MD, PhD, to make his presentation. Professor Tiribili, time is yours to give a lecture now. Thank you.
Well, can you hear me?
Yes, of course. We can hear you.
Thank you very much for this kind introduction. let me try to share the screen with you. That's already done. And so I can start my presentation. Let me tell you, it's really a pleasure to be a speaker of the 9th International Symposium on Innovative Bioproduction in Indonesia on Biotechnology and Bioengineering. What I'm trying to address today, I'm driving the presentation. What I'm trying to do today is actually make a link to what we are eating and drinking and the liver damage. As you know, no, it doesn't work. So why don't you remove my presentation? I'll share my screen.
Okay. Stanley, can you help me?
Yeah, I mean, I need to go on. Let me try to share my screen. And then, here we go. Okay.
So it's moved now?
Yeah.
Okay.
Can you see it?
Yeah,
okay.
I'm seeing it now.
Okay, let's move back to the start. Sorry for that, but okay. So liver is, after the skin, is the largest organ in our body, and in an adult weighs about 1.5 kilo. Although it's not that thick, the normal liver has a volume similar to the soccer ball. And during this period, we are very fan of a soccer ball because it's the largest gland in our body, and 80% is composed by the liver, by the hepatocytes. But most important, the liver is the organ in our body which is the largest number of functions. And liver can do more than 600 functions, such homeostatic, metabolic, detoxificant, and immunological function. But in addition to that, and not only a marginal issue, is that the liver is the only organ in our body able to reproduce and regenerate. And this is a Prometeo myth is being depicted. The main causes of chronic liver disease are different things, such as diet, here, medication, viruses, and autoimmune liver disease. Moving from normal liver to usually fatty liver, which is increased presence of the fat in the liver, which can go on with the steatohepatitis and then go to liver cirrhosis, which is advanced liver fibrosis and functional complication, ending up with the strip with the presence of hepatocellular carcinoma, which is the end-stage liver disease. We need to stop this trip from here to here. And what I'm going to address today mainly is the role of eating and the diet in having liver disease. So, and this is why, because MAFL, the so-called metabolic fatty liver disease, associated with liver disease, has a prevalence globally of more 25% of the population, which means in one out of the 4 European citizens has a fatty liver, so has a possibility to develop a serious liver disease. So if we go from the trip of from normal liver to hepatocellular carcinoma goes via different steps. First of all, we have the MAFL, the non-alcoholic liver, alcoholic fatty liver disease, which is mainly represented by the increased amount of fat into the liver cell. But once the inflammation occurs, due most probably to oxidative stress, we have a sort of activation of the extracellular matrix of the liver, which is able to create scars and then move from NASH to cirrhosis, which is labeled by the presence of scars in the liver, making the liver hard and less active. And these scars, together with the regeneration of the liver, end up in the hepatocellular carcinoma, which, as I told you, is the end stage of this trip. We have several risk factors for this. Of course, we have the overweight, the obesity. I'm going to touch this in more details later on. We have type 2 diabetes and insulin resistance. Altogether, overweight or obesity, type 2 diabetes, and insulin resistance make what we know now is called the metabolic syndrome. And the metabolic syndrome is related both in adults, but even more worrisome, is present also in children. And this as something we have to think about, because being in children, we need to prevent the fact that our child moves from here to here in the life span he or she is expected. And if we look here, that's very remarkable figure. That if you look the prevalence of obesity from 1975 to 2016, both in females and males, you may see the dramatic increase in the prevalence of obesity during this year. Meaning that the more so-called civilized we are, the more fat we are going to be. And this is something we have to cope with and we have to fight with. And actually, even more worrisome is the trends in the number of children and adolescents with obesity. And if you, both in males and in females, this is true all over the world. But if you calculate here, which is the Southeast Asia, also in the Southeast Asia, particularly in the male children are associated with an increased level of fat in the body. And if you look around the world, the presence of the metabolic fatty liver is basically almost the same all over the world. In some regions it's higher, like in US, in South America. In some regions it's lower, as Africa, due to the fact that there is a reduction in the calories available. Australia is basically like Europe, but in China we are observing now a booming wave of obesity, and this is something we have to cope with. So going back to what, in my opinion, is the most important aspect of the obesity is the so-called vicious circle of childhood obesity. We have a healthy child which actually due to a wrong life habits, sedentary life, lack of physical activity, is getting mildly obese child. And the mildly obese is actually a child is less prone to exercise, and those goes from mild to moderately obese child. And if this is also keep going with a wrong diet, is going to move from moderately obese child to severely obese child, and actually to and then to obese adult. And this is where we are going to go from a healthy child to a potentially sick child with the presence of fatty liver. And if you look around, you see that genetics may play a role in obesity. And we know now that the contribution of heredity to the risk of obesity is 25%. And this variability is related to the genetic background of the different studies performed. So we need to understand that habits, in addition to genetics, is the main predisposing factors, particularly habits of parents living with the child. And we just published an editorial in an international peer-reviewed journal, it was APT, making clear that it's the parents giving us the wrong information how to behave, less exercise and much more sedentary life. What are the targets of fat accumulation in the body? Of course, obesity is a multi-organ and multifactorial disorder. We can have damage in the liver, in the brain, in the heart, in the vessels, in the nervous system, and in the pancreas, and in the muscles. Altogether, meaning that we have a lot of complications of obesity in the childhood. We have damages in the behavior, psychosocial, neurological, like pseudotumor. We have pulmonary, sleep apnea, asthma, and intolerance of the exercise. Of course, cardiovascular, due to the dyslipidemia, hypertension, coagulopathy, gastrointestinal, as I told you about steatohepatitis, and gallstones, renal, endocrine, and muscle. So this, we have to conclude that an obese children, like an obese adult, are sick, and we need to cope with. So which are the main causes? It is the imbalance between the calorie taken and the calorie expended. I mean, I'm used to say that the weight is like our bank account. If you eat, I mean, if you gain a lot of money, you don't spend, you get rich. But if you eat a lot of calories, but you spend a lot, you get poor. So we need to understand that globally, there's a striking decrease in physical activity. 1 in 5 boys and 1 in 10 girls is not having a real and appropriate exercise. And even more worrisome is that the amount of fruit eaten by our children is drastically reduced as compared to the ideal amount. Only 30% of boys or roughly 40% of girls eat a fruit a day, in spite of the fact that fruit is readily available all over the world. And the major suspect is sugar. Sugar, the white sugar, it's really being associated with the presence not only of obesity, but also liver disease. And the question is how much is too much? And we need to understand this because we can give advertising. And here is what we have. And what we have here in the slide is coming from the World Health Organization, telling us the content of teaspoons for each beverage. And mind that the WHO recommends consuming no more than 12 teaspoons per day. If you drink a Coca-Cola, you drink it's like you eat 8.5 teaspoons of sugars. If you eat a yogurt drink, it's like 7. But if you need an energy drink, you have 10. And of course, telling you that what we are using to give our children, the so-called sodas, are then maybe very dangerous. And here's what we have in as comparison. Of course, the blank is the water. There's no sugar. But if you look at Sprite and Coke, which is widely used, we have that 33.7 and 35 grams of sugar per each can. And even worse are Fanta and Schweppes when we go from 33 to 43 and 44 gram of sugar. So telling us that, I mean, we are not surrounded by a friendly beverage consumption. What is the total fat? The total fat should not exceed 30% of the total energy intake. And we have different kinds of fat. We have trans fats, the dangerous one, due to the so-called junk food. We have the saturated fat, which is related to coconut, palm oil, and alimentary, polyunsaturated skin, meat, and fruit. And we have then the unsaturated fat, which are the real good fat, that should be much higher than 90% of the total fat intake we have every day. And this is important to prevent additional damage. How the excess of fat?
Sorry, Professor Triboli, I may interrupt you because your time is only 4 minutes again.
My time is still 5 minutes.
Oh, 5 minutes. Okay, 5 minutes again.
I mean, according to my watch, it's still 5 minutes.
Okay.
So I'll be on time. Don't worry.
Yeah.
So how the excess of fat and sugar impact the liver? This is a cartoon. It's a very complicated cartoon. I've had no time, unfortunately, to go through. But the key issue is that insulin resistance and increased intake for the diet bring together the possibility to accumulate fat in our liver. And this is what we have to deal with. So do we have animal models to study the juvenile fatty liver disease? The answer is yes. This is work we have done in our lab, driven by Sylvia and Natalia and Pablo. And what they did, they reproduced the fatty liver, a juvenile fatty liver, in a mouse, giving the mouse a lot of fat and a lot of calories. And what we have, what they have been able to reproduce is a fully scaled presence of fibrosis, meaning telling that this may be a good model to deal with in order to understand if there is any good treatment to work with this. In addition to that, we know that fatty liver is really linked to a leaky gut, because the gut is leaving alone with a lot of toxins moving from the gut to the liver. And this is what we have observed, that in large intestine, we have a leaky, the presence of a less tight junction as compared to controls. So in conclusion, we can say that excessive sugar and fat since young age induce obesity-related multifactorial events that lead to progressive multi-organ dysfunction, fibrosis, leaky gut, altered glucose homeostasis, which means diabetes. And this is, and we have to realize that the progression in children is much faster than adults. So we need to take care of this aspect for the future generations. And we know now that in the future 20 years, all the liver transplantation will be related to fatty children at the moment. So we need to take action on that. So let me skip all this because I have no time here. Let's try to deal with the future perspective. We heard that insulin resistant, type 2 diabetes, and obesity is the accounts for metabolic syndrome, which is a booming of obesity. And to date, we are still missing something, but we need to do something as a diagnosis, screening, and efficient treatment. And the clock is still ticking. So thank you for attention. I think I did my job in time, and I used all my 20 minutes.
Okay, thank you very much, Professor Tribili. Thank you for giving us such an informative and interesting presentation. And ladies and gentlemen, now are the time for the question and answer session. We have 20 minutes for Q&A session. If you have question for both speaker, please raise your hand and or you can write the question in the chat room. Okay, I think we have wait a minute. I need to see. I think Miss Atika, are you ready to give your question?
Thank you, Dr. Yeah, my question is to Dr. Yvonne. She's gone. No. Yeah. Okay. So,
Yvonne, Yvonne, are you still in here?
I'm still here. I'm here. Yeah. All right. Okay. So my question is a bit practical, no, but I never done the analysis that like what you did. So my question is like how long actually to get from your analysis of synthetic gene cluster until the analysis of the mutant, if I'm not mistaken. It's like you did like cloning for all this experiment, right? If I'm not mistaken. So ideally, how long until you get the results?
Thank you. Yeah, thanks for the question. That's actually hard to say because it's mainly dependent on the strains to manipulate. Actinomycetes are not so easy to genetically manipulate, and depending on which kind of organism you have in hands, this really depends how fast you are. But concerning the whole progress or process. Depending if you have the genome sequence, this is the first step. Let's say if you have the genome sequence, then you could go for the anti-mismatch analysis, and this is quite fast because you just upload the sequence on the anti-mismatch tool, and then you directly get the output after a few minutes. But this is, let's say, pure bioinformatics. It can give you an estimation what kind of gene clusters may be encoded. But you then need to really have a closer look into that and study that more in detail and also manually. And then, I mean, nowadays we have these overexpression plasmids in hands, they are cloned. Then you just need to introduce them into the actinomycetes. And if everything works well, you are maybe successful after 1 or 2 weeks. If it's a strain that is harder to treat, then it can take several months, I would say. But this is what where I skipped a slide, because what I wanted to mention is that nowadays we have different genetic tools. So we have different plasmids, which can be used either for protoplast transformation, which is a process how you can get DNA into the strains, or you can use the plasmids for conjugation, where you use the opportunity that you have an E. coli strain that can deliver the plasmid to the actinomycetes.
Okay, Fon, thank you for the answer. And in the chat room, I have 3 questions for Professor Tribeli. First question is, it is true that some herb like curcuma is help in maintain our liver health? Thank you. This is from Mr. Elok. Miss Elok, I think. Are you ready to answer?
Yeah, the answer is maybe. I mean, there's no real confirmation that any drug is preventing liver disease. There are some drugs used to or suggested to be helpful, but I mean the real scientific demonstration is not yet. The curcuma is one. The other one is silymarine, the thistle syrup. Others are the extract from the apple. But I mean, there are some information, but not real specific and solid scientific demonstration. So we need to take this with a very, with caution.
Okay, thank you for the answer. Okay, and the second question is, what are the most recent and reliable treatment to deal with liver damage that are already on cirrhosis stage? Is surgery and transplantation are the only option?
Well, it depends about the stage of the disease. Of course, if you have an end-stage liver disease, transplantation may be an option. Actually, it is an option. We have to realize that in Europe, every year we are performing roughly 2,500 liver transplantation all over Europe. But I mean, the ideal is not to go to the liver transplantation. The ideal issue is to stop the trip before moving ahead. This is what I try to demonstrate in my presentation as far as obesity is concerned. If we start changing our lifestyles by asking our adolescents and ourselves to exercise more, to eat less, and to be much more pragmatic in terms of daily behavior, that could be very helpful. Not only for the liver, but also for the cardiovascular disease, for diabetes, for everything.
Okay, thank you for the answer. And next, are there any possibility to recover from any damage in the liver?
For what? For?
Are there any possibility to recover from any damage in the liver?
Absolutely. I mean, liver is a regenerating organ. If you have a acute hepatitis, I mean, and you heal from the hepatitis, I mean, like A hepatitis, the liver returns back totally to normal structure. So we don't have to have any scar. Of course, if you don't recover fully, for instance, from B hepatitis or C hepatitis or other diseases, then the situation is not acute, it's going to be chronic. So the damage is chronic. So the end result is that the liver is daily damaged by the heating stimulus. And this ends up with something which is chronic liver disease.
Okay, thank you for the answer. And now we still have 13 minutes left. If there are any question for both of speaker?
We have been too clear.
Okay.
Or too confused.
Okay.
I have no idea.
If there's no any question again, I will ask maybe to Professor. Okay, Professor Mas. I want to ask Professor Iwan, what medium was used for self-activation with genome sequence in DSMZ strand? Is there any effect of the medium on the successful use of the self strategy in activating the silent gene cluster?
Yeah. Actually, also here we tested a different set of cultivation media just to make sure that we do not miss anything. So actually we tested 3 to 5 different cultivation media. Some of them you may know. I will not go in detail with these ones, but it turns out that if you have activation, that 1 or 2 cultivation media are enough. So it looks like that really the abundance of the subtype activator is more relevant than the media you use.
Oh, I see. Okay.
Thank you,
Yvonne. Yeah,
but Ira, I think I also see a question in the discussion panel or in the chat room
I will check
from Hatati. I may also read it by myself because the question was what kind of data that one inputs to get biosynthetic gene cluster using Plantis mesh. So I'm not sure if this is if I understand the question correctly, because there are different tools available. So Antismesh is the program that can predict antibiotic biosynthetic gene clusters encoded in bacteria, and Plantismesh is a tool where you can identify gene clusters from plants. So if this question really targets Plantismesh, and the question is which kind of biosynthetic gene cluster one can find there, I would say that you get an output which mainly contains gene clusters encoding for terpene substances, type 3 polyketide gene clusters, alkaloids, and so on, because these are more substances or gene clusters that are more abundant in plants. In bacteria, you may get just another output also on gene clusters encoding for non-ribosomal peptides, type 1, type 2 polyketides, for example. So I hope I could answer that question.
Okay. Yeah, thank you, Yvonne. Is there any question again for the both speaker? Please raise your hand or write it in chat box. Maybe I will ask Professor Claudia Terribili. Does consuming too much fiber also causes liver damage?
Yes. I mean, the point is not what you consume. The point is what the amount you consume and the calories. I mean, if you eat, let's say, a kilo of bread, the amount of calories is much higher than a kilo of salad. But of course, the taste of 1 kilo of bread is much better than the taste of 1 kilo of salad. And if you put some butter on your bread, the calories increase. So I think we need to make a balance of our daily behavior. I mean, if we eat more calories, we need to spend more calories. As I told you during my presentation, our weight and our metabolic functions is like the bank account. I mean, if you gain a little amount of money, but you are so able not to spend, you're getting rich. But if you gain a lot of money, but you spend a lot, you spend more, you get poor. So everything is balanced. We need to be careful. And then the basic point is to teach our children to behave properly. And our children, at least in Europe, don't behave properly. They are in front of a screen. They don't move. They don't go to walk. They don't go to play football. They don't go on the bike. They stay home, and this is bad. That's why we're having a real problem with it with adolescents.
Okay. My next question is there any effect of a supplement on the liver health?
You mean the supplements?
Yeah, like vitamin?
Well, I mean, the supplements have never been demonstrated to be helpful. The supplements are very helpful for the companies that are making the supplements. They're making a lot of money they're making a lot of good for the producers of the supplements, but there is no evidence that are making good for the liver of the subjects taking the supplement.
Okay, thank you.
So take a message, don't take supplements.
Oh, okay, thank you. Thank you for your suggestion.
Eat a fruit, it's more than enough.
Oh, I see.
You're lucky enough to live in a country like Indonesia, where the fruits is so beautiful. And you have mangoes, you have everything.
Yeah.
Why don't you have to take supplements? If you're living in Europe, I mean, you go and then eat an apple or an orange or a pear, and that's it. It's better, it's cheaper, it's much more healthy. Trust me.
Yes, of course. Thank you for your suggestion. Okay, if our time is left 2 minutes. Okay, or Yvonne, do you want to ask the question to Professor Tribelli?
Yeah, actually, I would also want to ask a short question to Mr. Tiribelli. I wondered if there are any numbers. What is in general more harmful if one drinks too much alcohol, or using too much calories from early days on?
I didn't touch alcohol for obvious reasons, because alcohol, there is a safe amount of alcohol daily, which around 30 grams per day, both in females and males. 30 grams of alcohol per day means half a liter of wine or a small glass of super alcoholic schnapps or whatever it is, which means that if you have one glass of wine with your meal, it's good. What is bad is to drink outside the meal, because the absorption of the alcohol without any aliment is very quick. This is why the heavy drinkers are used to eat together with alcohol something which is oily, like olives and peanuts, because you have a sort of film of oil in your stomach, so the absorption is less, and then you can drink more. That's the trick they're doing. So, a limited amount of alcohol is safe, actually. But, it depends on how limited is limited. And, a good glass of wine is good. And let me tell you that, unfortunately, I'm a teetotal. I cannot drink alcohol, not because I don't want, because I cannot stand it. And this is my limitation, because, with a good meal, a glass of wine, assuming that your religion is allowing to do it, it's good.
Okay, thank you for the answer. There's 3 questions again in the chat room. First, for Professor Tribelli. The question is, as we are getting older, is it true that our metabolic rate are also slower than when we are young?
Yes. Yes, the answer is yes.
Yes, okay.
That's it.
Yeah. And there's 2 questions for Yvonne. Did you also conduct any research in Germany on the culture to activate gene cluster for the production of natural products? Based on your experience, which substances from terrestrial and marine ecosystem produce more new metabolic at this time?
Okay, concerning question 1, we didn't do co-cultivation approaches, and I tell you why. Because later on, the identification of the produced metabolites is just too hard. If you do co-cultivation, you may activate something, but you do not know what is produced, and then you need to go again into the lab, isolate the substance, do chemical analysis to really identify the produced substance. We had the attempt to do a targeted activation, because if we use these kind of regulators, then we exactly know where they bind, which kind of antibiotic gene cluster is activated, and this facilitates later on the identification of the compound. And therefore, we didn't do co-cultivation, which also may work, but we only focus on this targeted activation approach. So the second question is, which kind of organisms do produce more antibiotics, terrestrial actinomycetes or the marine-derived ones? We cannot say until now. You can have marine organisms with really a lot of antibiotic gene clusters, but also terrestrial ones can harbor more than 50. So I don't see a difference in terms of the potency of terrestrial against marine organisms. So there's still enough out there to be identified in terms of new secondary metabolites, I would say. Okay, but I think there's an additional question. Eva, would you like to?
Okay, I think there is no any question again. Okay, I think its time is finished. Time is the end for the question. Okay. Finally, we come to the end of the seminar. I want to draw a conclusion from what speaker said. From Professor Ivan Mas, we can learn that a self-guide strategy can be used to activate antibiotic biosynthetic gene cluster in a targeted manner and thereby exploit the biosynthetic potential of actinomycetes. And from Professor Tribuli lecture, we can conclude that excessive sugar and fat consumption from a young age can lead to the obesity, leading to the development of metabolic associated fatty liver diseases and non-alcoholic fatty liver diseases in the children. Unfortunately, the development of metabolic associated fatty liver diseases in the children is faster than in adults, and efficient therapy to treat this disease doesn't yet exist. Hence, the best options still depend on the lifestyle changes. Okay, finally, I would like to thank to Professor Ivan Mas and also Professor Claudia Tribuli for their informative and interesting presentation, and all the participants for their very active participation. And now I will hand over the event to the MC. Thank you. Ms. Aulia, time is yours now. Thank you.
Terima kasih.
Terima kasih banyak.
Thank you, Professor Tribuli, Professor Ivan Mas, and also Dr. Ira for the amazing and also fresh sessions this afternoon. Distinguished guests, ladies and gentlemen, now we have arrived to the end of today's conference. On behalf of official committee, I would like to thank you for your attendance and also active participations on the first day of ISIBio 2022. We're signing off. See you on the second day of 9th ISIBio tomorrow at 9 AM Jakarta time with another interesting speakers and also materials. Stay safe and also healthy.
Bye-bye. Thank you.
Thank you very much, Yvonne and Professor Tribuli.
Thank you. Bye.
Bye.
Bye-bye.
Bye-bye.